So we'll start with our students. They're going to give us a presentation. We'll have an opportunity for you to ask them questions about their work, and we've got 3 of them. We'll start our student presentations with Muhammad Farooq, and he's going to be talking to us about a very relevant subject, avian influenza, what is holding it. Now, Muhammad Farooq comes to us as a third-year PhD student at the Kareem Lab at the University of Calgary. Before pursuing his PhD, he worked as a veterinarian for 5 years, providing vet care for both pets and poultry. Now, the Kareem Lab research focus is on avian viruses, particularly studying their pathogenicity and investigating the host immune response. So we're looking forward to what his research is telling us about AI. And let's put our hands together and welcome Muhammad Farooq.
Good morning, everyone. And thank you so much for the introduction. I think it's okay. And thanks, David, for the nice talk. And it was all about AI. And I'm also going to talk about AI, but unfortunately, it is a kind of different AI, which no one wants to hear. It is the HPAI, the eye pathogenic avian influenza. And it is actually about our new project that we got. We are looking into the environment, that what is actually holding the virus. There are a lot of materials, a lot of matters in the environment, but we just want to look and make connection. Where is actually the virus hiding and how it gets into the barn? And I'll just move into the news. The AI is in the news, especially from the states, and it is not from the poultry. It is in dairy cattle. More than 85 herds have been noted. They have found positive for the AI, and it has been found in more than 10 states. And another alarming thing is 4 people have been found positive for the H5N1, and they've got exposure from dairy cattle. That's a kind of alarming thing that everyone is looking into, especially the WHO, that how this is coming from the birds into the mammals. We used to listen a lot of mammals like the foxes and other wild mammals, but now it went into the dairy cattle. So it's not, they are thinking it is not far away from humans. And we already went through a pandemic, so no one wants another pandemic. And for the poultry in the state, it has affected a huge number of birds. 96 million birds have been discarded due to the AI outbreak. And about livestock or dairy in Canada, we are fortunate or we are unable to detect it. Up till now, no dairy cattle case has been reported from Canada, but the CFIA is testing the milk frequently and also taking the samples from dairy farms to see if the AI is affecting the dairy cattle in Canada. I'll also go into the background of the outbreak here in Alberta. And it was first on April 6th that a commercial poultry farm was detected infected with the iPath avian influenza. And it was the outbreak number 11 in Canada. And then it carried on, continued. The last farm, commercial farm that was detected as a positive was on February 19th. And this is the map of Alberta bordering Saskatchewan, and you can see the areas of the gray areas. Those are the revoked region, and 2 regions still are the control zones. So most of the gray zones are in the western part of the province. And when it comes to the number of birds, not as much as in the states, but still around 11 million birds have been affected in Canada. Second, when it comes to the outbreak, there have been 82 outbreaks noted in Alberta. So what our project actually is, first of all, we wanted to know how the farm was infected. Did the virus come from the here or it came from the water or from the wild birds. And the second objective of our project is, once there is an infected barn and the CFIA comes to do the procedure of depopulation, they gas the barn. The gas is then released in the environment. We are really interested whether the gas that is released in the environment contains an active and infective virus or not. So we sample the hair, and how long does that virus travel? Does it active, able to infect other farms or not? That is the second object. The third objective is, there are a lot of non-migratory and domestic birds like sparrows, pigeons, crows. Do they also contain the virus? Are they playing any role in transmission of the virus from around the farms or not? So what kind of sampling we do? We visit the farm when there is an infection outbreak, and we are regularly in communication with the team CFIA. And when we visit the farm, it can be any farm, a layer farm, a broiler, or a turkey farm. And then the CFIA also has this role of 1 kilometer vicinity. When a farm barn is infected, they try to depopulate any farm that is within 1 kilometer of that barn. So we take the oropharyngeal and the clinical swab from the infected farm. We also take dust sample from the infected farm, and we also then look for the any dead birds, that is the non-migratory bird or migratory birds or domestic birds from that area. And we also look from the water body near that barn, which we usually find, and take water sample from that one, and also take the water sediment from the from the area where the water body is. We also take the any dead birds from near the vicinity of the infected barn. And we are collaborating with NCFAD lab in Winnipeg. They have biosafety level 3 lab, and they have very well established protocol for the HPAI diagnosis, for the virus isolation and virus propagation. That's the point where we come to know, when we get the virus in our sample, we want to know whether this is a replicating virus, an active virus or not. So there comes the role of NCFAD. And they have also the sequencing facility, which help us linking the origin of the virus, whether the virus that is found in the barn or in the wild bird or in the water or in the hair sample, are they all the same or different? And is it note the way the hair blows? And we are also interested in that. This is the bobcat air sampler, and I want to let you know, this was extensively used during the COVID era to detect the COVID in hair samples in different areas, in different vicinities like airports or populated areas. And here we are taking the sample from our air sampler. This is our water concentration pump. So the best thing about this is, it concentrate, the filter concentrate the water. It converts 100 liter of water into just 50 ml. And then we look for the virus in that 50 ml of the water. And again, we are setting the weather station and air sampler near an infected barn. In the background, you can see the team CFI with their vehicle. And team CFI from the West operations are with all their members at an infected farm. They maintain the biocontainment zone. So whenever they visit the barn, the infected barn, they maintain 3 zones. There is a hot zone where no one can go, but with the proper PPEs, a transition zone where the hot zone people move to the when they are moving to the cold zone, and each and everything going into the barn is disinfected twice. And in this picture, you can see there are a lot of other things involved near the infected farm. The domestic birds, in the very deep background, you can see a cow standing there. And the fecal material of the migratory birds and also the non-migratory birds that I just mentioned, that we are interested to sample those as well. And up till now, what we are getting in our results. So our results are based on molecular technique, the PCR technique. We didn't look for these samples for the live virus, but we have sent the samples because we are not able to process this virus in our lab to test whether the virus is live or not, active or not. But we only test it with the PCR molecular technique. The oropharyngeal swab, glycal swab, and dust sample from the infected farm were highly positive. Similarly, the dead birds found near the infected one, they were also positive. And interestingly, the here before ventilation that we did, we found that positive for influenza viruses, but that was not particularly H5. So it can be any flu virus. We didn't check further. But the year after ventilation also was positive for the H5. And the other samples that we received, we checked was the domestic bird fecal swab. They were negative for the virus. The migratory bird fecal swab, we didn't find any positive RNA of the virus. And also we didn't find the RNA of the virus in the water sediment. About the water sample, we still have not tested it. It is under process to be tested. And our future plan is to stay connected with the CFI and follow the outbreak, collect the samples, the air, water, and all the other environment sample that I just mentioned. And also, we we are thinking whether the virus that is coming is spreading from the here or, and also we are looking into the venting procedure. Does it has an active virus or not? And also the water sample will be processed to see whether the water is a source of retention of the virus. One more thing we want to do is visit in off season the outbreak farms and see whether still that water or sediment or other environmental sample contains the virus or not. And another thing is this is from the infected farm, the one that I showed data. We will also be doing all the sampling and procedures in control farms. So we are going to select control farms. For that, we need your cooperation, the producer. They are already cooperating with us. And I want to thank them for the water quality project and for our bioaerosol project. And also for this HPI project, we need cooperation to visit for the control farm. So it's a kind of non-invasive. We'll just be doing the sampling and there won't be much the biosecurity threat. And with that, I would like to thank the members of Kareem Lab and also the people in NCFAD, the West operation of CFI. They have been very helpful and very cooperative, and all the other people in the University of Calgary, our funding agencies, Alberta Chickens, Egg Farmers of Alberta, and also the poultry veterinarians and all the people, they are collaborating with us in this project. And I'm also thankful to Western Poultry Conference for for taking the lead for this conference and inviting us. I'm more than happy to take any questions if you have.
Well, thank you, Mohammed. Obviously, it's a very a hot topic right now because we've got plenty of questions coming in. We'll start with what cycle threshold value is generally used for a positive identification of the virus in a given sample. So again, what cycle threshold value generally used for a positive ID of the virus?
Okay. So we are following the criteria of the CFI. They have set 35 CT value as a positive when there is a sample. 35 CT value or threshold is set as a positive in qPCR.
If you do find a positive sample on a farm, I'm guessing you mean a control farm, do you have to report it to CFIA?
I don't think on a control farm we are going to report to CFI. And because the positive sample report that we get From CFI, we are not rather telling the CFI. We get the reports from CFI.
Okay. Based on your research and current results, what do you think might be some key steps that farmers should take to keep the virus outside of their barns?
Yeah, it's again like a big topic of the biosecurity. So biosecurity is the main thing to keep the virus outside. And mainly it is thought about the wild bird. It is the wild bird that are playing their role, their spillover. So to just take care of that, and whenever there are wild birds, the best thing is to avoid the wild birds, like contact to avoid the contact the wild birds.
So no contact. Now, here's an interesting one. Is there any data collected on flies picking up the virus in the fields and bringing it into the barns?
Very interesting. We were first also interesting to take the beetles and the flies and the insects from the barn, but unfortunately, we tried in two barns. We couldn't find any of them. So it's interesting. We haven't looked into that, but there are reports. The people have found the low path virus in some of the bugs. They are taking it mechanically, like on their body. It's not like the virus is replicating in those bugs, but it is mechanically they are transporting it.
That's the ultimate biosecurity if you can keep flies out too. Wow. What is the brand name of the water concentration filter that you mentioned?
Oh, we take it from the Enovec. I think I just don't exactly remember, but it is from the States, and it is being used for virus sampling specifically. The filter size is important, that it filters all the other stuff but retains the virus. So yeah, it's interesting.
All right. Domestic wild bird fecal samples were not positive for HPAI in your testing. Was there any evidence of domestic wild birds being infected, such as dead birds?
We were not able to find any dead domestic birds around the infected barn. And there are some reports, people are looking into it, because I saw some people finding some dead crows, dead owls, dead pigeons, but we were not able to spot any. We were just picking the droppings of those birds randomly. So that's why, and checked those were not positive for the HPI.
Have you seen any positive testing without symptoms?
Well, because we have not done the control farms, when once we do the control farms, that will be the case if we find some positive. Otherwise, from the infected farms, I have this data only.
Okay. And then will you be doing this study in a more in a more condensed area, such as the Fraser Valley in BC? Or I guess, are you cooperating and seeing how your results match with other provinces, or are you the only one doing this kind of research?
So yeah, it's again interesting. We had a meeting with the people from Quebec and Ontario. They're also starting similar kind of projects which they've not started yet. Not sure about BC, but this our project is only for Alberta. It is not for a large range.
Okay. And then, so I guess, are there areas in Alberta where there are more kind of condensed farms? Is that where you're hoping to get some control farms?
Yeah, exactly. Yeah, that's what we will need cooperation from the producers.
All right. And what do you think is the future of AI control? Do you think it will be vaccines?
Yeah, there is another conference like syncly going on in the US. It is about zoonotic. So I heard about the vaccines, and especially in the France, they have done vaccine in ducks, and it is proving to be positive results. Like they're not getting that much infected. The transmittability is low. So maybe the US and the CDC, they think, who are importing the vaccines are starting vaccinating the birds.